Myself, I am Gunakar, Assistant Professor of Zoology, Government Degree College, Siddipet, Telangana State. Today, let us discuss prawn reproductive system. Reproductive system or reproduction is an important characteristic of all living organisms, and it is the process by which an organism produces young individuals of its own species to maintain the continuity of race. Animals generally undergo two types of reproduction: asexual reproduction and sexual reproduction. And the asexual reproduction can be seen only in lower organisms. Sexual reproduction is seen both in lower organisms as well as higher organisms. So the prawn undergoes sexual reproduction only. The prawn is a unisexual animal and shows sexual dimorphism. That means male and females are clearly shows differences. So what are the differences? Let us see. And before going into details of sexual dimorphism and reproductive system of prawn, let us have a look at the systematic position of prawn. Prawn belongs to the phylum Arthropoda because of having jointed legs, subphylum Mandibulata because of the presence of mandibles, class Crustacea as it is breathed by gills, and its habitat is aquatic habitat. It belongs to the subclass Malacostraca because the body is divided into many segments and each segment bears appendages. It belongs to the order Decapoda because of the presence of 5 pairs of walking legs. It belongs to the family Palimonidae because of the presence of rostrum with teeth or serrated rostrum at the anterior end. Belongs to the genus Palimon and species Malcolmsoni. Sometimes it is also called Macrobrachium malcolmsoni. Its common name is freshwater prawn. Now coming to the prawn reproductive system. As I said earlier, it is a unisexual animal. That means it shows sexual dimorphism, and male and females can be seen clearly, can be differentiated clearly by some of the characteristic features. So first and foremost characteristic features that differentiates male and females is generally the male is bigger in size than the female of the same age. So you can see in the figure, this is the male one and this is the female one. So female one is generally smaller of the same age of when compared to the male one. Now coming to the other feature. So if you are unable to differentiate just because of the size, so let us go to the next characteristic feature, that is abdomen. So you place the two male and female animals before you to differentiate the animal and see the abdomen. See the abdomen from the ventral view, and if you see the narrow abdomen belongs to the male prawn when compared to the female. So abdomen of female is somewhat wider when compared to the male prawn. Then these are the two differences. Now even then you are unable to differentiate, then let us go to the next characteristic features, that is the base of thoracic legs are more closely arranged in males. Base of thoracic legs are more closely arranged in males than that of the female. So now here in the female, thoracic legs are somewhat distantly arranged. So this is the third characteristic features if you want to differentiate male from female. Now even then you are not able to differentiate after the three characteristic features. Let us try for fourth characteristic feature. What is the fourth characteristic feature is the second chelate legs. Second chelate legs are longer, stronger and more spiny in males than that of the female. So second chelate legs are smaller in female, second chelate legs are longer in male and even stronger also, and has more spines and setae than in the female. So let us see what is second chelate leg. See, so this is the second chelate leg. Now you can see here. So this is the second chelate leg. So you can see here, second chelate leg of female is somewhat feeble, that is not stronger, whereas the second chelate leg of male is somewhat stronger, looking stronger. So I think with this characteristic feature, we can differentiate the male one very easily with that of the female one. If you are unable to differentiate from first three characters, this characteristic feature, that is strong second chelate legs, give you easy identification of the male one from the female ones. Now even then, if you are unable to, or to confirm that you have selected prawn is male, then go to the next characteristic feature, that is appendix masculina is present in second pleopod. Appendix masculina is present in second pleopod. So this go for the which is absent in the female. So in the pleopods are nothing but the appendages of thorax. So go for the appendix masculina, that is present on the second pleopod. A small, the tube-like structure, a small tube-like structure that is present attached to the second pleopod. With the help of that appendix masculina, you can clearly identify the male one, where appendix masculina is absent in female prawns. To confirm the male characteristic features of the male prawns, then you go for the another two features. They are the epimera of abdominal segments are smaller when compared to the female. So epimera of abdominal segments in the female are larger. So this is the only characteristic features which are larger in females, whereas all the other features are somewhat larger and bigger and stronger in male prawn. Now coming to the last feature that will confirm us from the male prawns from the female prawns is the genital openings, that is male genital apertures are present on the coxa segment of, that means the coxa is a part of the fifth pair of walking legs. So the coxa, that means so fifth pair of legs, if you see the coxa of fifth pair of legs, you can see inside male genital apertures are present on the inside, and it is membranous coverings. So you have to watch them clearly, and that means very keenly, and only they are visible. So again, but here on the fifth pair of leg, if you want to observe the female genital apertures in the female prawn, they cannot be seen because the genital apertures of the female prawn are present on the third pair of legs. So hence this clearly differentiates. The last characteristic features clearly confirms us that by observing the genital openings on the fifth pair of legs, they are confirmed that is the male prawn, and whereas if the genital openings are female genital apertures are present on the third pair of legs, if you observe that genital apertures are present on the third pair of legs, then you can clearly confirm that it is a female prawn. So with the help of these characteristic features, that means the prawns are not only unisexual, but they show sexual dimorphism very clearly. Now coming to the actual reproductive system, prawn reproductive system. Gonads. Let us start with the gonads. Gonads means here the reproductive organs. The reproductive organs in prawn, either in the male or female, they show some similarities. What are the similarities? Let us see. Gonads are similar in position. That means they are similar in position, shape, size, and general disposition in both sexes. In both sexes, gonads, their position or their location in the body, and then shape in the body, shape and size. All these, the general disposition. This disposition means general looking in both the sexes are same. So they show dissimilarities. Just now we have seen as they sexually dimorphic, but gonads in some features they show some similarities, that is in position, in shape, in size and general appearance. Both the sexes, the gonads are alike. So now you can see here this now you can see here this one and this one almost same. So you can see on the upper part of the slide, you can see the same position, same shape, almost same size and general appearance in both the sexes are same. Now coming to the where they are present, the gonads in the prawns are present in the region of thorax, posterior region of the thorax, above the hepatopancreas and below the pericardium or pericardial sinus. Below the pericardial sinus, where the heart is located, below that, and above the hepatopancreas in the posterior part or posterior region of the thorax, the gonads are located in both the sexes. Then they extend anteriorly up to the renal sac. They extend anteriorly up to the renal sac and posteriorly up to the first abdominal segment. So here its position, its shape, size and general appearance of the gonads in both the sexes are alike. That is, gonads in the male prawn are called testis and the gonads in the female prawn are called ovaries. So both testis and ovaries in the respective sexes are similar in position, similar in shape, similar in size and similar in general appearance. Now coming to the details of the reproductive systems of both the sexes. So prawn, as you know, whose scientific name is Palimon malcolmsoni, male reproductive system. So what are the main organs that are present in the male reproductive system and the main organs in female reproductive system? Let us have a look. Now you can observe this here also, almost the same same shape, same size, same appearance, they look alike. Now you can have male reproductive system. Prawn male reproductive system comprises of one pair of testis, one pair of vasa differentia. Vasa differentia is the plural, vas deferens is the singular, and seminal vesicles, one pair of seminal vesicles and one pair of genital apertures, male genital apertures. Now these seminal vesicles, otherwise called as vesicula seminalis, so where they are present, let us see. Now testis, as I said, there are one pair of testis are there. The two testis are soft, white and elongated bodies, and they fuse at their anterior ends. So the male reproductive system, which has the testis. So a pair of testis, they are whitish in color, though color appears in this figure differently. They are actually whitish, soft and elongated bodies, and they fuse at their anterior ends to form a common lobe. So they fuse at the anterior end only. Here they are fusing here, but at the posterior end they are separate. They are fused at the anterior end only and they are separated at the posterior end, forming a common tube-like structure and lobe-like structure. They enclose in their middle a small gap now you could see this here, a small gap can be seen, and this gap is meant for the passage of the cardiopyloric strand. So this cardiopyloric strand is meant for attachment of the heart to the pyloric stomach. So that passes through this gap. The cardiopyloric strand passes through this gap in between the two testes, and that's why the gap is left between the two testes in the middle. Now each testis consists of a large number of coiled, narrow and thin-walled tubules called seminiferous tubules. So seminiferous tubules, let us see later on in another figure. Now let us have a look at the vasa differentia. Later on, let us go to the structure of testis, internal structure of testis. So vasa differentia. Now this vasa differentia, you can see here very coiled structure, very much convoluted structure. A narrow tube, a long coiled and narrow tube, which is called vas difference. And I can say this is the vas difference. This is one vas difference and this is one vas difference. I already said it is singular. If you want to say it singular, you should say it is vas difference. But plural is vasa differentia. And this vas difference, each vas difference arises from one testis on either side, one testis. So the vas difference arises from the posterior end of each testis. See here, you can see convoluted, that's why. So which starts from the posterior end of the testis. And on emerging out, the vas difference of each side at once forms a much coiled mass. See here, very much coiled. Just now after coming out, immediately it makes a very much coiled or convoluted tubule on it, convoluted or coiled mass, and then runs ventrally downwards. Runs vertically downwards. See here, runs downwards vertically, and between the abdominal flexor muscles on the inner side and thoracic wall on the outer side. So they run downwards vertically between the abdominal flexor muscles on the inner side and thoracic wall on the outer side. Then coming to the next part, which is the seminal vesicles. They are already, I told you, one pair are there. So you can have a look here that the end parts are posterior most part of each the vas difference has a swelling like structure. Swelling or a bulge like structure, which is called a tube like structure or a swelling. So those are called seminal vesicles. So on reaching ventrally, near the basis of the fifth pair of legs, each vas difference swells into a club shaped structure, which is called seminal vesicle, otherwise vesicula seminalis. So you can call it as vesicula seminalis or seminal vesicle simply. To remember easily, it is better to remember as seminal vesicle, into which spermatozoa are stored in the form of white compact bodies called the spermatophores. So in these seminal vesicles, the spermatozoa are stored in the form of a spermatophores. Now these seminal vesicles are meant for storage of spawns or spermatozoa. Then each seminal vesicle open to the outside or exterior by a male genital aperture on the inner side of the base of the coxa. We have already mentioned here, we have already discussed that in the differences of male and female, that the male genital aperture is present on the coxal part of the fifth pair of walking legs of its side. So each male genital aperture is covered by a small flap of integument also. So that's why you need to observe carefully for the visual of for the to show or to see the male genital aperture, it is present below the integument. So uh, you have to keenly observe for its appearance. Now just now we have seen a pair of testes are there. So a pair of testes are present or attached at the anterior end and not attached at the posterior end, leaving a small gap for the passage of the cardiopyloric strand. And then from the posterior part of each testis there arises a small tubular structure which is called vas deferens, and this immediately when it comes out, vas deferens are very much coiled and make a coiled mass, and then runs downwards vertically up to the sixth up to the leg on the thoracic wall, and then on reaching its just near the thoracic wall or leg, it swells up to little extent, forming a seminal vesicle for the storage of the sperms, and then it opens outside in the coxal segment of the coxa of the fifth pair of walking legs, outside through a small aperture which is called as male genital aperture, that is covered by an integument. So far we have discussed. Now coming to the female reproductive system. Now female reproductive system, like that of the male reproductive system, also consists of another gonads. Like the gonads of female reproductive system are called as ovaries. So here male reproductive system gonads are called testes, whereas female reproductive system the gonads are called ovaries. A pair of ovaries are present and one pair of oviducts are present, and like this as the male reproductive system possesses seminal vesicles, here for the storage of eggs there is nothing is there in the for the storage of eggs. So here female genital aperture is present, so one pair, they are also one pair. Now let us have a look at here before going into the female reproductive system. Let us have a look at the detailed structure of the testis, inner structure of the testis. As I said earlier, the two testes are soft, white and elongated bodies, and they fuse at the anterior end of the ends to form a common lobe. They enclose in their middle a gap for the passage of the cardiopyloric strand. Also I have discussed already. Let us have a look at the internal structure of the testis. Now each testis consists of a many number of coiled and narrow and thin walled tubules which are called as seminiferous tubules. Sometimes they are also called seminiferous lobules. Now these are the lobules or tubules, seminiferous tubules, many seminiferous tubules. They are externally they are covered by the cavity of these tubules is covered by the single layered epithelium which is called germinal epithelium. And these epithelial cells, and that means these tubules are held together at their places by dense connective tissue in between. Here you can see this is connective tissue in between. So this connective tissue keeps the lobules at their place, and each tubule is lobule is surrounded by each lobule cavity is surrounded by a single layered germinal epithelium. The epithelial cells, they undergo spermatogenesis. That means they are some of the germinal epithelial cells they remove from the layer and they detach from the layer and then enter into the cavity and they undergo the spermatogenesis. Then they after the spermatogenesis they form the spermatozoa. Here so inside the cavity, so they detach from the germinal epithelium and enter into the cavity and they undergo spermatogenesis to form a spermatozoa ultimately. Now you can see here the mature sperm, how it looks like. This is the mature sperm. The sperm has three parts. Now the uppermost layer is the uppermost part is the head region, where the head region is a globular, almost globular structure which lodges a crescentic nucleus, a moon shaped, half moon shaped nucleus, and then having a blunt tail also. So having a blunt tail here. So this is the structure of a mature sperm. So this the spermatozoa or sperms, they produced within the seminiferous tubules. The mature sperm is looking like this. Now coming to the female reproductive system, just now I told you the female reproductive system has one pair of ovaries and one pair of oviducts and one pair of female genital apertures. Now you can have a look at here in this figure. So in this, these are the ovaries, two ovaries. You can differentiate them with a small gap in between them. That means two ovaries are attached to the posterior as well as anterior ends closely, leaving a small gap for the passage of the cardiopyloric strand. We already discussed in the case of male. In the same way, in the females also, the ovaries, they leave a gap in between for the passage of the cardiopyloric strand that holds the heart in position in its the pericardial sinus. And the ovaries are white. They are also white, though it appears in different color here in this figure, but they are white, compact and sickle shaped, sickle shaped almost. They are almost sickle shaped structures here. So here like this, sickle shaped. Okay, so this sickle shaped, and bodies touching each other at both ends, but I already told you, leaving a small gap for the passage of the cardiopyloric strand. The shape and size of ovaries vary with the age and the season of the year also. So the breeding season of the prawns generally is May, extend from May to July. May, June, July. These 3 months is considered as a breeding season. So depending on the season also, the shape and size of the ovaries also are changed. But whereas in the case of males, the shape and size of the testis will not change. Only in the female, the ovaries will change. Each ovary is enclosed within a membranous capsule and is made of numerous rows of ova. That inner structure we shall later on discuss. So okay, the immature ova lie towards the center while the mature ova towards the surface of the ovary. See the a small oviduct. See here in this figure also you can see these are the oviducts. This is one oviduct which is coming from this ovary, and this is another oviduct which is coming from the same position of another ovary. So short and wide and thin walled tubes, the oviducts, originate from outer middle border of each ovary. See, outer middle border, not like that of the vasa differentia which originate from the posterior end of the testis, but here the oviducts, they arise from the middle part of the ovaries, and then they run, each oviduct runs vertically downwards. Here you can see each oviduct runs vertically downwards to reach the coxa, to reach the female genital aperture on the inner side of the coxa of the third walking leg of its side. So here, so you need to understand and you need to remember this. For the female genital aperture is present in third walking leg of its side, whereas the male genital aperture in the male prawn is present on the coxal segment of the fifth pair of walking leg. So this is the clear differentiation that can help us to differentiate male and females easily. Yeah, now coming to the prawn. Now male reproductive system, it is clearly seen completely again. Then let us have a look at once again, and then go to the next part. The testis, as I said, is very much soft, white and elongated bodies, fuse at anterior and posterior ends, leaving a gap for the passage of the cardiopyloric strand. Then each testis consists of many seminiferous tubules embedded in connective tissue. Cavity of tubule is lined by a single layer of epithelium. Then cells of epithelial cells undergo spermatogenesis to form spermatozoa. And mature sperm consists of a rounded head, nucleus and tail. Just now we have discussed by seeing in this figure. Then vasa differentia, I told you plural form of vas deferens is vasa differentia. Long coil tube arises from the posterior end of each testis. It runs downwards to the fifth pair of leg to open outside. Then seminal vesicle, a small club shaped structure on each vas deferens, stores the spermatozoa in the form of spermatophores. Opens to the outside through male genital aperture on the coxa of fifth walking leg. Now this is the matter that belongs to the prawn male reproductive system. Now coming to the female reproductive system, we have to glance it over it. Then female reproductive system, as I said earlier, the main organs are ovaries. They are a pair of white compact sickle shaped bodies touching at both ends. They leave a gap in the middle for the passage of pyloric strand. They enclosed by a membranous capsule. And generally the ovaries consist of radial rows of ova in various stages of development. And now in this besides figure you can see here this cut portion of this each ovary now, and immature ova at the center and mature ova on the surface. Then mature ova are nucleated with plenty of yolk. Then the oviducts originate from outer middle border of each ovary, as we have seen just now. Then they run ventrally downwards. They open by a female genital aperture on the coxa of third walking leg. So just now we have discussed that the breeding season extends from May to July. So the breeding season of the prawn extends from May to July, May, June and July. And during the breeding season, the male and female prawns come near, and then the fertilization happens. The fertilization is external. So the male deposits sperms near the genital openings of the female. So during this closing of the male with the female, the male deposits sperms near the genital openings, that is on the third walking leg, near the genital aperture, female genital aperture. Then eggs are fertilized when they come out. So eggs, there is no storage of eggs, but the mature eggs, they come out during the breeding season through the female genital aperture. As and when they come out, they are fertilized, because already the male deposits the sperms near the genital openings. So here eggs as and when they come out, then they will be fertilized. Then female lays eggs in large numbers. See, the female lays eggs in large numbers because generally the animals have to continue their race. So as the environment is not always same, it is always changing, and it poses many problems to the organisms to live. So that's why to withstand and continue their race, generally the prawns are also, generally many animals, they produce in large number. They produce their young ones, or they produce their eggs in large numbers. In the same way, the female prawn also lays eggs in large numbers, which are attached to the pleopods by a sticky secretion of tegumental glands. The female is now during the breeding season, female is now said to be in berry condition and carries the fertilized eggs wherever she goes. So at that time, then it will attract the male one, and then the fertilization will be continued. Then development is direct. That means there is no larval stage. So young ones come out of the eggs after 4 to 6 weeks. They cling to the swimmerets of mother for some time. Then after a series of molts, they grow into adults. Usually the prawn, it lives about 3 to generally 5 years. So only the thing you have to remember this is, during the rainy season or during the floods, many eggs or matured eggs or the fertilized eggs will die, because they cannot withstand the speedy flowing water. So they will die. So if at all, if some of the eggs, some of the fertilized eggs, they sink to the bottom, they may live, or they may withstand the fast flowing water, but when they are subjected to fast flowing water, they will die. Most of them die. So generally, they're within the 4 to 6 weeks of the after fertilization, the young ones will come out, and the young ones will appear in the same way like that of the adults, but only in the differentiation is only in the size. So they undergo many molts, a series of moltings to grow into adult. And generally the lifespan, or the life of the lifespan of the prawn is generally 3 to 5 years. Thank you.